biology, topic by topic. Today we're going to have a look at plant defences, animal defences against pathogens, and then we'll have a look in more detail at probably what goes on inside a human. Let's have a look first at plants. Plants here, if we just zoom in here, are basically rather an odd way of trying to deal with a pathogen. Basically, their first problem is that we've got some sort of pathogen coming in, and this pathogen is going to affect a plant cell. Now, the plant therefore needs to sort of recognize that it's not just a plant cell that's being attacked, but in fact lots of things are going to happen. So plants not really very passive in this respect. They in fact respond quite well. So there are in the plant cells here receptors, and these receptors respond to molecules from the pathogens, and they produce something which can then go to tell other plant cells to sort of be prepared, I suppose. So we get various signalling molecules, and these signalling molecules try to sort of give out the information. There we are, a bit better to see. So let's have a look and see what's happening. And if I draw out a plant cell, and typical plant cell, we've got some sort of cell wall, and somehow this pathogen, whatever it's going to be, is going to find its way into the cell. So as soon as this is basically trying to penetrate the cell, then we've got something happening. Now, we've got some molecules from this pathogen will be recognized directly by the cell here. I suppose we can put in here our nucleus, and basically once this is happening, we've got here in the cell membrane, we've got these receptors. These are going to sort of give some indication that we're having some damage. Therefore the cell's going to do something about it. So basically this nucleus is going to pick up these messages, and they're going to produce new chemicals, and those are going to be callose and lignin. Now these chemicals are going to basically strengthen the cell walls to try and make them better. Now callose basically is polysaccharide. It contains beta-1,3 linkages and beta-1,6 linkages between the glucose monomers, and this is basically going to happen within minutes of the initial attack. Callose is synthesized and it's deposited between the cell walls of So if I draw this out as another cell coming along, then we've got basically something here. There's going to be another layer there going on. We've got something sort of starting to fill up this, and that's Corlos doing that. This basically acts as a barrier, and this basically tries to prevent pathogens entering the plant cells. Large amounts of this stuff continue to be deposited in the cell walls after any sort of viral infection. Lignin is then added, making a more of a mechanical barrier, and that makes this even thicker and stronger. Corlos can block things like sieve plates in the phloem, sealing off the infected part, and this Corlos is deposited in plasmodesmata between infected cells, sealing them off. So we're thinking then this is acting as an effective barrier, trying to sort of make the cell thicker, harder to get to. But plants are well known for their chemical defences. So chemical defences, what have we got? Well, let's think of some of those. The obvious chemical defences are against insects, and we've got various things like pine resin, lemongrass produces a nice chemical here called citronella. There are in fact against insects various chemicals like chrysanthemums. So chrysanthemums are going to basically produce a chemical called a pyrethrin. So chrysanthemums, these are wonderful plants to grow. I'll see if I can remember how to spell this. Yeah, that's right. Chrysanthemums are wonderful plants. If you're a gardener and you've got lots of veg, you put your chrysanthemums amongst them, and these chrysanthemums, they produce all these pyrethrins. These pyrethrins come along and they basically kill the insects. So it's quite an effective insecticide. This is in fact here a neurotoxin. Oops, neurotoxin. So it's quite wonderful. And in fact other plants make things like caffeine, and that is really toxic to insects and fungi. But we also know that against bacteria we've got various chemicals that we've met. We get some plants that produce some phenols. This certainly doesn't do bacteria any good at all. We've got a chemical called glossipol. And that's made by the cotton plant. And that doesn't sort of do an awful lot of good to bacteria either. There are various plant proteins that disrupt the bacterial and fungal cell membranes. We've got our favourite, lysozymes in our lysomes, the organelles which break down all the cell walls. We've got to deal with the antifungal systems, and as I mentioned, we had the phenols. This doesn't do these things any good, but there are lots of interesting things that some plants produce. Chitinase. Now this doesn't have any effect on the plant, but it breaks down the chitin in the fungal cell walls, so that's not going to sort of do the plant any harm, but it's certainly going to do an awful lot of harm to the fungus. There are also some more perhaps general toxins that plants produce, and these vary in various different things. Cyanide is sort of quite a common one they make, and you probably know about the castor beans producing ricin, and that certainly doesn't do anything any good. So plants' more passive system, there is this system with collos that really at the moment I don't particularly understand how it works reading the literature. I don't think really anyone got a really good idea. We know what it's doing, but how still waits to be sort of seen. So plants generally a passive system, and one way they also work is just to have general things in them, like stuff that if you're going to eat them you're going to die, so don't eat them. Animals. What do animals try and do? Well, our first job, I suppose, with an animal, let's try to basically keep out the pathogen. That's going to be our first basic idea, and that means skin. Skin here is basically a barrier, and it's going to prevent the entry of pathogens. If we look at your skin, it's absolutely covered in pathogens. The idea is that if you can actually on your skin keep quite a lot of healthy, friendly bacteria growing there, there isn't room for pathogens to start getting in there. And you also produce sebum, which is an oily substance that inhibits the growth of many of the pathogens. But unfortunately we've got holes in the system, so we've got to deal with the entrances to the body. The first of those would be the eyes, and I'm always cheered up to know that, you know, oh, that tear, it's so romantic and lovely. Yeah, it is, until you start going into the biochemistry of it. And here we've got chemicals like lysozyme, which literally chops up any cells that sort of get in there, be they bacterial, fungal or whatever. Far from being sort of a romantic, lovely thing here, this is nature at its cruelest. and most effective. Your eyes are ideal. They're warm, they're wet, they're behind it, I suppose, dark, and it's made of food, the four things that all organisms are looking for. Yet with that hidden lysozyme, everything dies pretty quickly, and getting eye infections relatively rare. So that works quite well. Our other problem is, of course, other things. So there you can say we've got lysozyme, then we've got the mouth, the nose, not the ears, and the anus, and basically I suppose we've also got, women will have vagina, or in males I suppose it is just the urethra. So what have we got to try and deal with these? Well, basically mucous membranes. We've got sticky mucus. This will simply trap bacteria, and in the mouth and the nose they're basically on their way down to the stomach. The mucus also contains lysozyme, which certainly won't do it any good. If we look at urine, that also contains lysozyme, and so that's going to have quite a sort of effect on anything getting in. If we think about the anus here, it's absolutely full of all sorts of flora here, but basically it's an exit, and if things are going out, then it's not going to sort of do things much good. It's hard to sort of infect going in when lots of stuff's trying to come out. Vaginas are more of an interesting problem, and that is fairly acidic and also contains other chemicals in it which basically trying to kill off any sort of infections getting in there. So the entrances and exits to the body are fairly well covered. The big problem that we've actually got is other things trying to get in. So we've got various expulsive reflexes. What do I mean by those? Well, very easy. We have the cough, we have the sneeze. Both will try and eject pathogen laden mucus from our system. We've also got vomiting to expel stuff from our stomach and mouth, and we've got the system of making diarrhea to expel the contents of the gut, plus any other sort of infective pathogens we've got there. So these are effective for us. Of course, all of these can then cause infection in other people who are nearby, but it works well for you, and basically keeping you alive is the first priority. So our next problem comes with wounds. Now, we've got problems here. As soon as the skin is breached, the pathogen's going to enter. So we've got various things that we've got here. So the first one is bleeding. Basically, if it's coming out, it's going to be hard for something to go in. And then immediately we have clotting. And this system of basically sealing the wound works very effectively. So let's have a look and see what happens. So here we're going to hit some damage. We've got some damage to some tissues, and that is basically going to activate the platelets. And what these will do is they're going to release a chemical called thromboplastin. What thromboplastin's going to do, it's going to meet up with prothrombin, and in the presence of calcium ions, then this prothrombin is going to be catalyzed by this thromboplastin to make thrombin. And then the thrombin here is going to take fibrinogen, which is a soluble thing, a soluble chemical. It's going to catalyze that, and that's going to make fibrin. These are little fibers, and that is going to form the clot, and with the platelets, they're going to hold the platelets in. So we're going to have a system where we've got some fibrin, and then the platelets are going to fit over the top and that's going to seal the wound. It's quite effective. Once the clot dries out, it forms here a scab. Once we've got that, now the underneath the epidermal cells here, these are going to reproduce, and that is in fact going to take this temporary seal and give us a permanent seal. And while that happens, we're also going to get our blood vessels will here grow, stroke repair, and once they're done, then we get collagen fibers being sort of deposited to strengthen the system, and when the epidermis reaches normal thickness, then simply the scab falls off. and you're better. The wound is all healed. So that works particularly quite well. Now, the next one is an inflammatory response. An inflammatory response is we've got some sort of localized here response to a pathogen. And here you're going to feel pain. Often we're going to sort of see heat in a tissue, which we'll often see as redness, and quite often swelling. So these are the sort of things we're going to see. And special cells that we have called mast cells, these are going to be activated. Now, what do these mast cells do? Well, they release chemicals, and the ones that you're probably familiar with are the histamines. And the cytokines. Right, so what do these do? Well,
sorry, I don't know that, but I do have a
Alexa,
like.
Stop. Good. Always encourage when it helps. Let's have a look at histamines. What do they do? Well, they make the blood vessels dilate. Now, that's going to basically cause the heat of the system, the redness, and that is simply doing things like raising the temperature. Now, raising the temperature in a wound is great because this is going to effectively destroy the pathogen. So that's good. Now, the histamines, as well as making the blood vessels dilate, what they're also going to do is they're going to make them the blood vessels are going to become more leaky. And as they become leaky, so basically we're going to have the blood plasma is going to leak out, and that is going to call up a swelling, which, posh word, we call this an edema. And that, surprisingly, causes pain. The other chemical that's released the cytokines, and what they do is they simply attract white blood cells. Specifically the phagocytes, and they basically dispose of all the pathogens by using their process of phagocytosis. And if this infection is more widespread, then you might see this simply as a rash. So this is our inflammatory response, and quite effective it is too, but it does cause quite a few problems, and people sort of complain, oh, I've got this, that and the other, and they think, oh, I've got a spot, and they want to try and burst it, which doesn't actually do much good, because you're then getting rid of all the white blood cells, which are there basically fighting the infection. You're much better off leaving them. So we've got a fair number then of non-specific defences. So we've got to try and now get rid of anything that's actually got into the body. So we've got this idea of keeping things out, but now what happens when we've got something in? So we actually now need to get rid of any pathogen that's got into the system. Now there are two ways we can do this. We use the immune system for more specific things, but we've got to try and now get rid of anything with a sort of that's already got in, and we've got to try and get rid of this. So the ways we're going to try and do this is certainly the first one, which is a fever. This is a way of trying to get your body to sort of upset the little pathogens that are in there. So our normal body temperature is around about 37 Celsius, and most pathogens reproduce best at lower than 37. Higher inhibits the pathogen reproduction. So we make a fever, we take the temperature above 37, and this will basically inhibit the pathogen reproduction. And in many cases it's actually going to sort of work quite well. Also we find that our specific immune system works faster at higher temperatures anyway, so that's going to have a sort of a double effect. So we've got a fever. Now the next one is phagocytosis. Phagocytes are special white blood cells that are going to engulf and destroy the pathogens. There are two main types. There are neutrophils, and we've got our macrophages. Now, how are these going to sort of work and kill off anything? Well, basically we need to get these substances here, these cells, and we're going to need them at the site of an infection to attack the pathogens. And basically we want to try and kill off everything, and it's the cytokines that are going to bring these here. And when we look at things like pus in a wound, this is basically going to consist of dead neutrophils plus the pathogens. Yeah. Right, so let's see how these things actually work. So we've got the pathogen attracted here. So we know many of the pathogens here, they're going to produce some sort of chemical that's going to attract the phagocyte. So the pathogens have got here some sort of, on them, an antigen, and this will attract the phagocyte. Now, once the phagocyte is there, and let's just draw in here our phagocyte. Our phagocyte, it's got its nucleus, and it will have in it various bags, lysosomes. Now, somehow this phagocyte is going to sort of recognize this pathogen. Now, normally it's going to try and do this by a chemical scent. It's actually scenting the antigens and moving towards the strongest concentration of these, and it's looking at these and it's looking for something that is basically non-self. All your cells have got little labels on them that says, this is me, this belongs to me, whereas this pathogen doesn't have that. So these phagocytes then recognize this as being non-you, and what we're going to then do is we're going to try and do the engulfing of this pathogen. As it does so, it produces a little sort of small little vacuole, and this is called a phagosome, and in here is going to be our pathogen, not well drawn to size here. So we got this simple phagosome in there. Now we've also got in here lysosomes, and these lysosomes move towards the phagosome, and what we're then going to see is this phagosome is going to bind to the lysosomes, and all those chemicals are going to be released into here, and basically this is all going to be chopped up and destroyed. Now, what the body tends to try and do is to take any antigens on here and present these antigens on the surface. With the antigen presented on the surface, then we can now move to the next stage, where the phagocyte can now put those antigens onto the surface, and these then can be used to stimulate other cells to try and identify and produce particular things to stop this. And that's going to be our immune response, which we'll look at. The chemical here is actually called an MHC. This is a major histocompatibility complex. And this now becomes what we call an APC cell, an antigen presenting cell. And these antigens are now stimulating other cells in the specific immune response system to do something. And all of this takes place in about 10 minutes. So the system has to be quite fast to try and get everything to work well. Other things we've talked about fever, phagocytosis. We can also look at the blood cell count. You often see that when people aren't well, they have a blood test and they take a matter of some sort of cells and they try and have a look at seeing the number of red blood cells and white blood cells. And you've probably done this and had a go at trying to do this. So this skill of trying to sort of take a blood smear and then count it is really quite useful. If we then stain this, then you can easily identify between the white blood cells and the red blood cells, and we're looking at basically the white blood cells to count up how we're doing with this infection. It gives us an idea of how well we're responding to these. So that's our third thing that we're looking at, to see that the blood cell count will actually normally rise as we got some sort of infection. And I suppose the last one that we can actually do here, number 4, is you can produce various different chemicals. We mentioned earlier the cytokines, which are going to basically attract other phagocytes to the system, to this area where we've got some sort of problem. And we've also got some opsonins. These are chemicals that bind to the pathogens and tag them so they're more easily recognized by the phagocytes, and then these phagocytes then can engulf them easier. There are a whole load of these, but we've got quite a few of them, and they're known as the immunoglobulins. the two of the ones that are well known are immunoglobulin G and immunoglobulin B, called IgG and IgM, and these seem to have the strongest effect on what's going on. So this is all about the non-specific, the stuff that's going to happen all the time, that you've got all sorts of things. You've got the skin to protect you all the time, you've got your eyes, nose, mouth causing problems, wounds, big problems, and if somehow something gets in you, then you've got this general basic body defences. But how about something a little bit more specific? Well, next time we'll have a look at our specific system, the immune system. Until then, if you found this useful, then please subscribe, give us a thumbs up, and I will see you next time on A-Level Biology, topic by topic. Until then, stay safe. Bye bye.